Good afternoon. I have to start saying that I'm new in the field. My introduction into the ME/CFS research field started in 2010 when I joined the Catholic University of Valencia in Spain. Because one particular reason, which I think to today's audience will be of interest, and is that our dean has been treating patients, CFS and ME patients and fibromyalgia patients for over 20 years. So one of the things he's doing at our medical school is to make sure that every student that goes through the program learns about the disease. So he's doing an initiative on education of the future medical doctors to learn about the disease and to learn that there are many people that are suffering, that is not a rare case or any other thing. So, thank you. So, on the very first days I started there, my background is in molecular biology, and I'm a basic scientist, so I had never heard of the disease before he approached me, and he told me he wanted to take some action to help his patients. So, being from the molecular biology field, we discussed and suggested that a molecular approach to find differences, molecular differences in samples from the patients versus a good cohort of matched controls would be an appropriate design to look for these desired molecular markers. And then I put this up, which I think is a little bit on the line that Dr. Hanson just showed. So it's clear to everybody that if we reach the point in which we can find molecular biomarkers for ME/CFS, we will count with an accurate and rigorous diagnostic method that will accelerate diagnosis in the clinic. And not only that, these molecules that we hope we can identify, right, will provide the scientists with hints on what cellular cascades specifically are altered in the cells, and this should lead to learn more about the etiology and progression of the disease. Not only for ME/CFS this is true, but this is true for any disease, right? On top of that, and very importantly, biomarkers are needed as essential tools to find effective therapies in clinical trials and to develop appropriate preventive programs, because without those tools, there is no way to closely monitor how are these drugs affecting the patient, right? How are the drugs helping the patient? And the clinical trial may reach an end with no definitive conclusion. So after discussing, we decided to choose MicroRNAs. This is a small, we'll see in the next slide what are they precisely. But we decided to choose these microRNA expression studies in the mononuclear cells of peripheral blood of the patients for the following reasons. These microRNAs have been proven already be valid biomarkers for other complex diseases. Okay, they are also very stable compared with other RNAs, which are very labile molecules regularly, but the microRNAs, they lack a label on the 3' end. They don't have a poly A tail, and then that lacking that tail and the small size make them more stable, and so that leads to the possibility of analyzing even long-term archive samples. So that could accelerate if we go to biobanks and we can count with a larger number of samples, then that could accelerate also the discovery of a particular profile of molecules that could serve as biomarkers. So once they identified, let's say once it's known which microRNAs in particular are affected, based on those changes, it would be relatively easy to implement a diagnostic method, a simple diagnostic method in the clinic. And on top of all these reasons, we observed that this possibility was quite unexplored for the diagnosis of ME and other related diseases. So we took that decision. We were going to study what was that profiling. But before we go and see the results of the pilot study that we have done, I want to explain a little bit what are these microRNAs, which you can find in the literature also abbreviated as miRNAs or miRs. Okay, they are on average more or less 22 nucleotide non-coding RNAs. That means they don't make any protein, they don't make any other product by themselves. What they do is they are processed, they are expressed in the nucleus, transported to the cytoplasm, and after some processing, they bind to the target messages that are the RNAs that code for the proteins. The proteins are the effectors in the cell, right? So when the microRNAs bind to a specific region, the 3' UTR of these messages, the double-stranded is recognized so that the duplex induces the decay of these messages that code for the protein. So there are functions that are reduced, right? They are affected. And that is known to happen physiologically for all the time in all the cells of our body, and they are also present in all our body fluids, saliva, urine, plasma, and so on. So this is the title that you can find published last year in the journal PLOS ONE. And I'm going to tell you today what we evaluated exactly was the genome-wide expression profile of these microRNAs in mononuclear cells of the peripheral blood using microarrays. These microarrays are platforms in which we can find the complementary regions, so we can identify with which microRNAs are enriched or are decreased. Okay, as you know, you may know probably in Spain, we are our funding for research is quite short. So we were lucky enough to participate in an open call by these industries that was developing this set of microarrays, which covered the whole 1,212 different sequences, which was the complete sequences of different microRNAs known to be in the human genome at that time. And we convinced them that our study was important to do, so they decided to offer us the grant of evaluating 11 patients suffering from fibromyalgia with chronic fatigue as well, and population age matched controls, 10 of them. So this is the selection, the patient selection criteria, and as you see, they were all long-term sufferers, because we decided to start by this pilot study with patients that had been they had been diagnosed by more than one specialist, and they have been suffering for both fibromyalgia and chronic fatigue for a very long period of time. The idea was also that these long-term sufferers should show more drastic, different profile if this was present. This table only shows the demographic data of the participants in the study. And this table I'm showing only to illustrate that we made sure that the purity, which is set by these numbers here, that the purity and the integrity of the RNA of our preparations was optimal before being sent to Japan. We wanted to be rigorous, and in fact, the fact that the table shows these samples are of high quality doesn't mean that we only prepare this. We prepare more, and we chose from those the best quality to be sent for evaluation. And this is how it looks like, a heat map display of this, the results of this genome-wide microRNA screening. If we see, it's very small, I know, but you can probably observe a line that divides this set of signaling, right, of signals here from this right part. On the right, we have the healthy participants, and on the left, we have the patients. And as you can see, we could find many of them. The brighter colors, in fact, indicate lower levels of many of these microRNAs. As many as 16% we found, so of the 1,212, there were 193 that presented an inhibition of at least 50% in the patients. We try to look also for upregulated microRNAs, but in our pilot study, we didn't find any that was reliable. The very few that were upregulated were either expressed at very low levels, making us doubt about the validity, and with higher readings as for this one, this presented higher levels, but however, the inter-individual differences, the differences between one or other participant, both in the control and in the patient group, was quite particularly, changing. It was not. It was not replicated very well in either of the groups. So what this table summarizes is the ratios of the selected microRNA that were more drastically downregulated. And we can see that we selected these 5 top because they showed a ratio of at least 6-fold downregulation. And some of them, you see, even reaching over 13-fold. We also chose these 2 in the bottom part because being with a ratio around of 1, they could serve as a positive control of microRNAs that were not affected in either of the groups. So that means the levels were similar in the group of patients and the group of controls. And these are the raw data, the readings of these selected microRNAs to show you simply that the values were quite reproducible, quite similar between all the participants, and quite different from the group of the control group in almost every case. When we plotted these values to see if there was significant and they could provide a tool to differentiate the control group from the patients, we see that clearly that there was a big difference for every one of the 5 selected, with more than 6-fold difference. Not for the ones that are controls, that is what we were expecting, right? We had seen. And then there was one, in this case, that was downregulated in the microRNA, but these are the data from another technique, the results of another technique called Realtime PCR or quantitative PCR. We use this alternative technique to make sure to validate those data with the same preparation, with the same patients and control samples. So then at that point, we wonder if any of these 5 microRNA that we were proposing as a signature biomarker of the disease could correlate with any of the clinical symptoms. But we didn't find any direct correlation. This was expected, though, because the number of samples was very small to show any significance. So didn't mean much, but if we plot every one of the values for these microRNA 143 and 145, that, by the way, are encoded together in the genome, we see that this patient number 9 had a value of mental fatigue much different than the rest of the participants, and show at the time there was a correlation with a higher value, meaning that has a value closer to the control group. This is not what happens with the control microRNAs, right? So meaning that perhaps this microRNA could be associated, could be measuring the mental fatigue of the patients. We don't know for sure yet, but this would be a hypothesis that could be further studied. So interestingly enough, there were other initiatives, apart from ours, from other groups, that use cerebrospinal fluid of fibromyalgia patients in Sweden, and they found downregulation also of 2 of the microRNAs. We also found downregulated in PBMCs. So cerebrospinal fluid is quite different fraction than PBMCs, and they should not be expected to find any correlation. But the fact that there is this coincidence makes us think that this could be, real markers of the disease. Another approach that has evaluated the microRNA levels by another method in plasma also detected altered levels of the 143. And then more recently, there has been proposed another completely different signature of microRNAs, this time from a fraction similar to ours, PBMCs. Okay, and this is the group that has done the work. So we checked the article to see what could be the difference of not having a single coincidence. The first thing we saw is that they use another type of array of fewer targets, and the other thing was that they use a very common system to extract total RNA that is called trisol. This substance is a highly denaturant and is used to extract total RNA. However, it is known that this trisol introduces a bias on the small RNA, meaning that when you extract RNA with this technique, unless you have abundant, very large amounts of material, of tissue or cells, then the small size RNAs, especially those with a low GC content, are lost. And I'm presenting here the exact text that you can find in this reference, where a group of researchers that had published previously another study in the known Journal of Molecular Cell, they retract their results when they realized that they had used a substance that biased the results. They show a comparison of the kit that we use, the Mirvana, with this trisol. And I just put it as an example to see that in the case of these 2 microRNAs that are comparing, using the trisol, depending on the number of cells that I were using, the ratio could vary from 0.3 to 1. That means threefold differences. Okay. So what is the take-home message of these last things that I mentioned? And the important, I think, take-home message is that the researchers should use a standard operating, and I took the freedom of adding this another O, optimized procedures. So instead of SOPs, I put this extra O. Should be established not only for a satisfactory diagnosis and correct diagnosis, which is currently, it's highly debated. It's quite well known, what SOPs to use in biobanking of the samples. But then there is very little efforts trying to optimize and standardize the downstream applications. So all the rest of the efforts in CFS and ME research would make no sense if we don't do also standardize and these standard, these operating procedures. I'm very enthusiastic about the COST initiative that it has been announced today, because with the platform that will be created, I strongly believe that it's going to help to improve the implementation of these SOPs that would be ideal. Not doing so, and if every researcher applies different techniques that are not optimized, it would only lead to a waste of resources and an extended time for the patients waiting for the results. So, in summary, what we propose is that the signature we identify could serve as a biomarker, at least a starting point to develop the optimal biomarker-based test for the diagnosis of FM and perhaps also FM CFS and perhaps of other related diseases. However, before going to the clinic, we have to validate this not only by another technique, as we did, but also in a larger study group. We plan to do this by including geographically distant cohorts. What is the reason? The reason being that the environment and the lifestyle could affect also the microRNA profiling. So having this done in two different locations, it's for sure an important step to take. And we plan to do this with collaboration with Dr. Lubov Nathanson, that is here in the audience today, and that has attended the BMC6 this year, that works in a group of Dr. Nancy Klimas, that you may know, and Nova University in the States, and also with Dr. Alegre's group from Vall d'Hebron in the Hospital of Barcelona, that is also an expert in diagnosis of CFS-ME, using these SOPs. So lastly, I want to comment a little bit about what could be the meaning of this, the regulation of microRNA. And I cite here two pieces of work that show how viral infection can target the machinery for the microRNA production. So, in fact, chronic recurrent infections could potentially lead to defined MIRNOMs. This word means a combination of a certain set of microRNAs, okay? So, however, at this point, we don't know if the MIRNOM that we are proposing as a biomarker would be deregulated due to viral infections or to any other cause. I want also to mention that when cells are infected, or they are stressed by environment or by aging, the endoplasmic reticulum of the cell, that is the place where the proteins fold, becomes stressed. And this stress leads to downregulation, to the cleavage of also the precursor of this microRNA, connecting the stress in the cell and the aging with the biogenesis of the microRNAs. And finally, I want to introduce you to the dean of our medical school that I told you about here in the center, and the rest of the team, which is the nurse. This is an hematologist working for us, a psychologist, and the predoctoral student that already finished her thesis last year. So thank you very much.
Well, don't worry about the disappointment of things not always turning out the way you think it's turning out. Further research always throws it up, and I really appreciate your honesty in actually saying, you know, these things happen. Mm-hmm. Questions, please. Mm-hmm. Well, I asked you one yesterday. I mean, what about this thing? I mean, is microRNA the big thing because the gene gets reactivated again? You don't actually destroy the regulation of the gene. You destroy the protein, knock it down a bit, but the gene is still there and will make more fresh RNA. So you may, you know, be knocking the microRNA out or something, you know, somehow, or not making the protein, but back it comes again.
Well, it depends on what is the cause of the destruction.
Yes.
Because if it's, that is, for example, is not being expressed, they are transcribed by RNA polymerase II, yeah. And if they are not made in the cell, then that could also show as a downregulation, as a less abundance. However, it's true that in vitro assays, when there is silencing, when these microRNAs are used to silence genes, silence the gene, but the protein is still there, and depending on the half-life of the protein, yes, the effect might not be immediate. But it is also true that if there is a single microRNA that is altered and is constantly altered, it would be a good target, a good therapeutic target, because of the small size. Yeah.
Any other questions? Yes, sir.
One of the features which seems to be emerging from this whole day, and particularly in what you said, is that the immunological part of the genome is dysfunctional in one way or another. And you mentioned the role of viruses in rendering causing this dysfunction. Do you have a view on endogenous retroviruses as a potential cause for this dysfunction?
Well, yeah, this is a possibility that at the moment is not known, but it could be a possibility that the insertion of a viral sequence in the genome would alter the expression of other genes surrounding the site of insertion, or even at other sites. Yeah, absolutely.
So thank you very, very much, Lisa, very much indeed for presenting that.